Alzheimer's disease AD and diabetes mellitus, primarily tied to diabetes T to D, are two of the most pressing chronic diseases worldwide. Over the past 2 decades, epidemiological and mechanistic evidence has established a strong bidirectional relationship between the two, leading to the concept of type 3 diabetes as a descriptor for Alzheimer's. We previously reported on this crosstalk and its implications for drug development in 2022. Since then, there have been significant advances in our understanding of the pathophysiological mechanisms driving the AD diabetes nexus and new treatment strategies, particularly those based on GLP-1 receptor agonists, GLP-1 RAS, have shown promise in preclinical and clinical studies. This report synthesizes current understanding of the AD diabetes crosstalk and highlights recent advances to 𝗚𝗟𝗣𝗠𝗘𝗧𝗏 in GLP-1 RA-based combination therapies, currently in clinical trials targeting both metabolic and neurodegenerative pathologies. A pathophysiological crosstalk between Alzheimer's disease and diabetes. Both diseases share common molecular pathways, including insulin resistance and impaired insulin signaling in the brain, which contributes to tau hyperphosphorylated, a beta accumulation, and synaptic dysfunction. Chronic inflammation is also a hallmark of both conditions. In diabetes, low-grade systemic inflammation is driven by peripheral insulin resistance and is exacerbated in the brain via microglial activation and dysregulation of pro-inflammatory cytokines. Additionally, mitochondrial dysfunction and oxidative stress play a pivotal role. Energy metabolism deficits in neurons and pancreatic beta cells result in redox imbalance, which further promotes neuronal damage and amyloidogenesis. Finally, amyloid cross-aggregation between islet amyloid polypeptide, IAPP, and a beta may occur, potentially accelerating plaque and tangle formation. B. Clinical and epidemiological evidence. The link between diabetes and Alzheimer's is well established, with T to D patients exhibiting approximately 50 to 100% increased risk of developing AD. Notably, brain insulin resistance correlates with cognitive decline in both diabetic and non-diabetic individuals. Furthermore, cerebral glucose hypometabolism and peripheral insulin resistance are early and consistent markers in both diseases, suggesting a shared pathogenic process. GLP-1 receptor agonists, dual-action candidates. Originally developed for glycemic control in T to D, GLP-1 RAs have demonstrated neuroprotective effects in preclinical models of AD. These agents enhance insulin sensitivity in the brain, reduce neuroinflammation, modulate synaptic plasticity and neurogenesis, and decrease a beta and tau pathology. Notably, they achieve these effects independent of their glucose-lowering action. D. Recent GLP-1-based advances in clinical trials to 𝗚𝗟𝗣𝗠𝗘𝗧𝗏. Several GLP-1 RA-based combination therapies are in clinical trials, each with unique mechanisms and advantages. 1. Semaglutide Novo Nordisk. The Trailblazer ALZ-6 and EVOKE-1/2 trials are exploring oral and subcutaneous semaglutide in patients with early AD, both with and without diabetes. Interim analyses in 2020 suggest that semaglutide may stabilize cognition and improve hippocampal glucose metabolism, particularly in diabetic patients. 2. Tirzepatide Lilly, a dual GIP, GLP-1 agonist. Emerging evidence indicates improved cognition and insulin signaling in the CNS. A phase 2 study 𝗧𝗥𝗡-AD is ongoing 2023 to 2025 in a mixed population of AD-T to D patients, with primary outcomes on cognitive function and brain glucose uptake. 3. Cotadutide AstraZeneca, a GLP-1 glucagon co-agonist initially developed for NASH and T to D. It is now being evaluated for cognitive endpoints in metabolic cognitive syndrome, a condition characterized by overlapping features of diabetes and AD, in early phase 2 trials. This agent targets both systemic metabolic derangements and central lipid and glucose dysregulation, which are key in AD progression. 4. Triple agonists GLP-1, GIP, glucagon. Preclinical and early phase human trials 2022-2025 show synergistic effects on neuroinflammation, weight loss, and amyloid reduction. Companies like Novo Nordisk and Hanmi are leading development, with human AD trials expected by 2026. Strategic implications. These findings support several strategic implications. First, therapeutic repurposing of existing drugs for new indications can be expedited, particularly for comorbid T-to-D populations. Second, precision medicine opportunities exist through biomarker-driven stratification, such as using insulin resistance indices or CSF biomarkers to tailor interventions. Third, with the AD therapeutic market valued at over $10 billion and the T-to-D market at over $60 billion, dual-purpose GLP-1 agents represent a significant commercial opportunity. Finally, real-world evidence will be crucial to validate cross-indication effectiveness and safety. In conclusion, the intricate interplay between diabetes and Alzheimer's disease necessitates systemic, multifunctional therapies. GLP-1 receptor agonists and their next-generation combinations, particularly 𝗓𝗥𝗏 co-agonists, offer a promising therapeutic class that not only addresses metabolic control, but also ameliorates cognitive decline and neurodegeneration. Ongoing clinical trials suggest that incretin-based agent may revolutionize the management of these interconnected metabolic neurodegenerative conditions. Definitive data supporting this dual targeting strategy is expected in the next 2 years, potentially transforming the therapeutic landscape.